It now falls to me to introduce one of our keynote speakers. I'm delighted to have the benefit of a talk from Dr. Michael Hawrylycz. I've known Michael for a very long time at the Allen Brain Institute. He will be talking about the Brain Initiative Cell Atlas Network, or BICAN, and speaking about comprehensive multimodal atlases of the human brain. Now, Michael has many hats. He's a PhD in applied mathematics from MIT. He's an expert in computer science, image processing, computational biology, and genomics. At the Allen Institute, he is the director of Modeling, Analysis, and Theory Group, which, as the name would imply, is responsible for informatics, modeling, and data analysis strategies. And he leads informatics and data annotation efforts for the well-known Allen Brain Atlases, for which we are all internationally grateful. And as part of BICAN, he is developing multimodal atlases of the human, non-human primate brains.
Thank you, Alan. It's a great pleasure to be here, and I'm really grateful to see. I had the good fortune to be part of one of the original kind of organizational meetings of Healthy Brain for Healthy Lives, and it's amazing to see how productive it's been over the several years. You can see immediately I've generalized human to mammalian brains, and that will be a kind of I guess a reason to have some mouse work into our discussions here today, but we'll try to tie it back as much as possible. Also, I want to say that we can think of, I guess, this presentation as kind of a companion with Rachel Hofstadter's presentation. Would be a training kind of, more specifically training in the tools. And you may see some duplication between some of the topics, but it'll be reinforcing, and I'll be there to kind of support her in her works. You can either probably, as questions arise, you can ask either of us probably as we go. But so I'm just going to kind of give you an overview of what's happening kind of in the U.S. kind of Brain Initiative Cell Atlas program in general, and I'll kind of tie it in. I guess we can think of this as educational in general sense of the word, where informative as opposed to just let's say there's going to be a test or something like that. So the U.S. Brain Initiative was started actually 2013 under kind of Obama, the call. It wasn't the first time in the U.S. that there was sort of a call for a coordinated large initiative. That there was something actually under George Bush Sr. called the Decade of the Brain. Maybe I think probably Alan will at least remember this. And there were always big aspirations, and sometimes things come out of it, but it often doesn't come out quite to the extent that maybe the original hope was. But one of the amazing things about Decade of the Brain, it did start, is it largely kind of formed the field of neuroinformatics. I would say before the so-called Decade of the Brain, there really wasn't really a well kind of group of organized people that you would call neuroinformatics people, but it drew a lot of neuroscientists into the interests, such as David Van Essen, other people like that, who dedicated a lot of their work to that. The Brain Initiative started in 2013 or so, and the goal was somewhat of a different focus. The intent was a recognition in biology and neuroscience that it's really technologies that have been the drivers of a lot of our progress, and it's to sort of utilize and apply those technologies toward information discovery and characterization of brain activity and brain function. And so there's several aspects to this program. It's not just about, let's say, it spanned different things from kind of neuroprosthetic devices through treatment strategies for neuropsychiatric diseases to a major program, and actually the largest financial investment is in the attempt at the characterization of the components or structure of the brain, the cell types of the brain, and their circuits and interaction. And that program is called the Brain Initiative Cell Atlas Program, and it has had three kind of phases since 2000, sort of late 2013, and they all have the BI for a Brain Initiative. The first was called BICCC, which is this in the green right here. This phase here, which was about developing prototyping the technology, trying to show that high throughput methods such as single cell sequencing, such as certain kind of track tracing, other things could be done in a scalable way that would make atlas profiling sensible. And it was about 10 different awards and different people, they did it, and it was successful. And then the next phase was this phase 2, which was called the BICCN, or the Brain Initiative Cell Census Network. And that went from about 2017 to 2021 or 22, and that was basically large-scale profiling in the mouse with some prototypes in the human. And so it was like, the goal was a complete characterization of the mouse brain to the extent possible using these technologies. Finally, now we're kind of in this phase. In any event, this red, the phase that we're in now, this sort of called Brain Initiative Cell Atlas Network. Now, largely they changed you know how they changed the name of these things largely because they want a reason that they can stop giving money to one person and give it to another. Otherwise, you just call it the same thing. It becomes like, why couldn't I be part of that? So it's got to be a whole new funding mechanism, a new award structure, and everything like that. And so, but this last phase is about. Thank you. Try this one here. For that one arrow, but we got the arrow. Here we are now, where that arrow is right there. And that sort of shows you where we are in the process. Okay, so that's basically kind of the overall structure of this. What were some of this so-called Brain Initiative Cell Atlas program was the intent of kind of spans all of these, the major interest of all these BIC blah, blah, blahs. But the goal was that essentially the realization that what you really want is you want a sort of a characterization of like the parts lists of the brain to the extent possible. Sort of a unified kind of genetic map and catalog, transcriptome, epigenetic data, chromatin, essentially maps. You want, to the extent possible, a kind of taxonomy, cell type taxonomies across species that can be aligned. You'd like the understanding of the regulatory environment and what cell types, how they're controlled. You'd like a spatial description of cell types, the distribution of the patterns of cell types in the brain, and imaging kind of pipelines and tools that enable us to connect this back with anatomy. And manipulate things also to control using kind of genetic tools to control these. So you can see it's a very ambitious thing, but the overarching goal is really trying to get at the types, the components, and their connections and circuits in the brain. And so here is the kind of, this is like, if everything were to go well, you would imagine that this might be something like the kind of, the system here. You'd have these kind of cross-species atlases here. We can link in with, all these different things, Human Connectome Project, various disease consortia, the Human Brain Atlasing Map, Brain Connects, NIH Armamentarium, and BigBrain, hopefully. And that's, I think, part of the thing we would like to, I'd like to keep that in mind over the next few days, is that if we can try to see and identify, as we go through discussions, some of these synergies here that may be possible. And I believe certainly there will be. So just to back up a little bit, in time, is that back in 2012, which, how science progresses, is that what was 2012 doesn't seem very long ago, but like in terms of progress, it's sort of like, it's sort of a long way. You look at things that go into papers 10 years ago and you think, they would never be published today, it's kind of amazing, and that's how this works, right? The sophistication of analyses has dramatically escalated and changed, and expectations, and it's kind of amazing how progress works that way. This was an atlas that we did. It was one of the first probably large-scale kind of transcriptome kind of atlases of the human brain, which we kind of finished around 2012. It was 6 different people. It was two females and 4 males, not that bias only based on availability and not intent. But it kind of was a whole brain kind of profiling using microarray technologies from about 900 different locations of the brain, and all this data integrated into this sort of big platform. The data from cell type resolution, the data today would be seen as kind of coarse, right? Because you're grabbing kind of sort of parcels of the brain in different locations. But Actually, the atlas has been quite extensively used, particularly in the neuroimaging community, for which there's a resolution match, which is quite good, you know, that when you start to compare kind of relative gene expression based on this kind of parcellation, it's kind of intersection and reconciliation with fMRI and other kind of sort of more imaging-based studies is very good, and it's been used quite a bit in the human brain mapping community there. And we kind of aligned this at the time, working with Katrine with a Hawrylycz kind of atlas, so that this kind of became a kind of a way of comparing, you know, kind of some of the data between these two atlases. So this work that has been used quite extensively, and sort of it set the prototype for the first kind of atlasing of the human brain from a transcriptome. So I'm not going to talk now about this BICCN phase, right? You would recall this is what I had mentioned was this mouse phase. In other words, so the idea is the technologies were proven. Now we want to scale it up entirely to the whole mouse brain with prototypes in the human and certain other things. But we wanted to get sort of understand if we could do this in the mouse before dealing with all the other complexities of human. And later in the talk, I'll go into some of the human aspects. This BICCN was a large network. It was probably like 20 or so PIs, major PIs, using all these different kind of technologies here of kind of single-cell transcriptomics, epigenetics, attack-seq, methylation data, also tracing data, neural projection data, whole brain morphology, spatial transcriptomics, different kind of the patch-seq technique whereby you record and do morphological studies and electrophysiological studies from the same cell. And this matrix here kind of showed all the different investigators and all the different techniques and for what species and how they were doing it. And I'll show you how, I'll talk about where that data is a little bit later. But basically, you know, this is just an illustration of kind of some of the approaches here. Is that how, and what you sort of got out of them, right? The single cell transcriptomics give you a sort of a type of a map, sometimes hierarchical in the organization of kind of cell types. The epigenomics data give you kind of peaks indicating potential kind of regulatory sites and their relationship to transcriptomics. The spatial data gives you, really importantly and increasingly used now, much less so in the earlier phases, but now everyone really wants to do it because the recognition that the spatial distribution of these cell types is very important to their identification. Also, now increasingly EM data too, with the Kinnex project and the electron microscopy data for ultra structural kind of thing, and a variety of other techniques, right, for manipulating these things. And then, of course, computational data management and analysis, you know, things very much of interest to HIBALL and bring this information kind of together. The initial project that we did in this was, so this group got together. None of these people had really worked together before. You know, we kind of, we had this consortium, this was back around, started in 2017. And it was, you know, we wanted to do a first kind of project. And so it was thought that what we would do is we would pick a certain area. We picked, in fact, the primary motor cortex, M1 or MOP in the mouse. And the idea was that this was because this was an area that was roughly functionally homologous between mouse, marmoset, and human. The organisms that we had could be identified readily in each of these anatomically. And each group would kind of apply these techniques to the examination of cell types in these organisms, just of the techniques I just mentioned. And so it was just one area. The ultimate goal of BICCN was supposed to do the whole mouse brain, but as often happens, you don't quite finish. You spend a lot of your time doing a comparatively smaller part of it, but you do build up the infrastructure and the capabilities so that once you get your pipeline going, you know, you can do it. What you want to be careful though when you do projects like that. What we did is that you tend to call it we called it a mini atlas, and it was thought that we were going to sort of bang this out in like 4 months or something like that, right? Well, as is common, it takes the bulk of the funding to do the mini atlas, and then you're sort of and sort of with no cost extensions, you find yourself doing the whole rest of the brain, which did happen, in fact. But it's just that just beware, don't call something a mini atlas if you get started, probably not a good idea, but that's what we called it. So anyway, this was a set of papers, it all came together really well. It was this kind of integration of all these data sets. It was published in Nature as a package in October 2021, and you can get all that data and all those data sets there. A lot of the things that made that feasible in the mouse was a very kind of established and kind of high resolution common coordinate framework. I mean, when I first started working in this area, the term common coordinate framework is not really that old. It was sort of everyone kind of, it was one of those terms that everyone knew as soon as it was uttered what we meant by it, but it was never clear like when it started or how far back it went, But sometime over the last 20 years, somehow this term came into common speech, but it was, and it largely, of course, means a kind of a system by which a set of typically images with annotation and anatomy where you can address kind of the organization and the localization of the data spatially, typically in an informatic way through mapping, through access, through APIs. The Allen Mouse Reference Atlas was originally drawn by Hongwei Dong. He used to be one of the original people of the Allen Institute way back in 2003. And, he kind of basically went into an office and printed out these gigantic Nissl images on large sheets of paper and locked himself in an office and sort of drew on these, And we would to someone at that point, and I really didn't neuroanatomy was not something I had any real knowledge of in 2003. It was quite something amazing, and I can remember talking to friends over at like Microsoft, and they would say, I said, how is he doing that? I said, well, it's kind of voodoo. I don't know what he does. He just goes in and he sees these things and he draws them on the picture. And they say, oh, that's not right. You should send us the images. We'll use machine learning and we'll recognize the areas and send it back to you. Great, fine. So I talked to Hongwei. So we send some images over to Microsoft. And as this often happens in academic collaborations, that no one ever calls you back and says, we can't do it, actually. We're having trouble, You just never hear from him again, So we never did. But that was about it. And so we never heard from him again. And the reason is that there's a lot that goes into the neuroanatomy, of course, right? There's a lot of subtle things. And a trained neuroanatomist who, to some extent, is often, I would say the genomics people have a little bit of a they have a little bit of a feeling that somehow that the anatomists are kind of old school and that everything should be recognizable from the genes. But the fact is that neuroanatomists are incredibly sort of adaptable and utilize an immense amount of information and historical information also available to them in drawing kind of an understanding atlas context. But the important thing here is to be able to bring it into software, right, and to align these things. And ultimately, what we did was that we used kind of more high resolution tomographic 2-photon to really produce dense series, as shown on the far right there, in which you can get a near continuous series and at very high resolution at 10 micron resolution, isotropic resolution. And then from there, this can now be annotated using 3D annotation tools and things like that. So that, and this so-called Allen Common Coordinate Framework with the mouse really has really done pretty well in terms of its usage and application in a lot of different kind of studies in the mouse. And it formed kind of the place where we mapped all the BICCN data of the mouse into. It enables kind of studies like this, for example, using techniques like FMOST, right, where you do sort of brain clearing and then kind of viral tracing and things like that, and you can produce really kind of long range kind of connections in the brain and really get long range imaging, and then actually kind of reconstruct them and map them into the thing to see. And this is a collaboration here with Han-Chuan Peng, who used to be at the Allen Institute and now is at the Southeastern University in China in a kind of collaborative institution. So one of the things that this led to was this kind of taxonomic organization of the cell types of the mouse brain. And this is what we're looking at here, is this is a taxonomy of the mouse brain, of the primary motor cortex, all the different cell types kind of organized, you know, the neuronal, non-neuronal, GABAergic, glutamatergic, and how they break up by the kind of different things. And you see, and then we could do kind of the morphological studies and kind of to make associations there to the extent possible. And sometimes those associations are not as, sometimes they're more predictable than others, but it can be complex. And then you can get these, the important thing is that, you know, despite the complexity, you can see these same kind of structures across regions, across organisms, and different kinds of things. You know, once this whole paradigm and mechanism was set up, we could apply this kind of to the whole mouse brain. And so this was done, so we did this, and a couple different groups did this. We did this at the Allen Institute. Joe Ecker's group did it with epigenetic data at the Salk, with Bing Ren from UCSD. And so this was basically now essentially using all these techniques to do sort of, you know, single cell kind of transcriptomic profile across all the regions of the mouse brain. You can think of it as in some sense the granddaughter of the original Allen Mouse Brains Atlas, which was done in kind of a high resolution in situ hybridization, section by section. You pick a gene, you look at it in situ hybridization. Now this is molecular level single cell data, brain wide. And also together also using with the spatial transcriptomics, right? Where there, you know, now you're with the, you know, with the spatial methods, now you're using, these are kind of the descendants of in situ hybridization, where now you design gene panels and you can get combinatorial expression in situ to understand kind of what, from a panel of markers, which genes are expressed where. And this is extremely important for understanding the distribution and the mapping of types. And so what we do, we have these kind of correspondences where we can now map between the transcriptomics and epigenetic data and pull that through. This is just showing kind of work here from Joe Ecker's work of the epigenetic atlas. And you see, when you look at kind of the basic structure of the organization, while there's a lot of complexities, there's definitely a correspondence of what types are identified and how they are going. An ongoing work right now, an area of very fruitful investigation is, right, you know, how are the epigenetic programs driving the kind of the correspondence of these types and the activity of these cell types. And so this is a lot of advanced machine learning methods are going into trying to use sequence level kind of data analysis of the attack-seq data toward prediction of gene expression and kind of identification of cell types. We put all this data into this thing called the Allen Brain Cell Atlas. I'm pretty sure that Rachel will talk a little bit about this. Yeah, she's raising her hand, and so, you know, I'll be there to support her when she gives her presentation, kind of, and we try to help walk through you some of this. But basically, so it's good that, I mean, we were talking early, and she said, well, what if we have duplicate slides? And I said, if we have duplicate slides, that's great, because people will be able to see, you know, get some reinforcement as to one thing, and nothing wrong with a little bit of reinforcement. But basically, you know, this is just kind of a way of navigating this data and going back and forth, and I'm sure we'll hear more about that. We have other visualization tools that we partner with lots of different people on. The Chan Zuckerberg Cell by Gene, a group from Leiden, Padua group developed Cytoexplore, and he's really made a kind of an industry out of a graphical visualization of transcriptomic and spatial data using kind of a basis of TSNE and other kind of maps and things like that. This data was all put into this, so we organized all this data, and one of the things we did at the Allen Institute was that we basically pulled all this data into a kind of catalogs, and we have a data catalog that sort of, you know, puts forth all the data, shows you where to get it. It doesn't have, its organization is not so robust that, like, you can go down and search across studies at a gene level or a cell type level. That would be great, but we don't have that. It's more of a project-based organization now, and you'd have to go to the publications in some sense to still kind of dig some of that out. But there's a data catalog. It summarizes by project, different things. You can filter the data in different ways and get different access to it like that. And there was a whole ecosystem which you will see that for dealing with this data, which in the mouse will be considerably simpler than what was necessary, and which I'll talk about for human data. But this was kind of this kind of more or less the organization of it. And one really salient kind of component of this, which was something that was which deemed essential is part from the NIH, was that they wanted rapid and regular data release, right? As soon as data was kind of produced, it should be QC'd, and then it should be deposited in kind of public archives where the public can get it. And this is kind of a way of making sure that the data goes out. Oftentimes investigators are reticent to kind of release their data, right? They think that because they worked hard, they wrote this thing, they raised the money, they generate the data, and they want their people to be able to profit from that and to write about that and to get publications which kind of, you know, help them, and that's all natural. But in reality, it's pretty unusual to be scooped by your own data. In fact, it occurs far less than people would imagine. There's a fear which is somewhat kind of unfounded in some ways. By generating the data, you have such an advantage of having actually constructed it, set it up. You've constructed the experimental design. I mean, you can be scooped. I mean, you'd have to kind of. It doesn't occur as often as people think, I think. So, but anyway, all the Brain Initiative data is in these different archives, and it's all kind of systematically processed. We had part of our work. We worked with a group from the Broad, Tim Tickle, and which they've developed kind of these kind of programs for kind of automated pipeline of processing the transcriptomic data, the multiome data, et cetera, the attack data. And most of that data, it all goes into this archive called the Neuroscience Multiomic Data Archive, or NEMO. And so this is all organized there. These are the other primary archives of the Brain Initiative data here. Is that the NEMO for the transcriptomic data, this one called DANDY from MIT with Satyajit Ghosh, stores electrophysiological, optical data, some behavioral organs things. The Brain Image Library with all our imaging data goes, including our spatial transcriptomic data. From Johns Hopkins, under Brock Wester, the so-called BOSS-DB, where EM data and various connectivity data, now with the upcoming Connect program, all that data will go there. And finally, Open Neuro for the MR and PET. So all of the data can be found in these different things. It's not cross kind of referenced as organized as well as one might like. And in fact, I have to admit a really kind of idiosyncratic and unfortunate side effect of it is that there's a technique called patch-seq, which I don't know, have you heard of patch-seq? Raise your hand if you've heard of patch-seq. Yeah. Patch-seq, yeah, it's a technique where you actually can, it's a really remarkable technique where you can profile multiple kind of assays in a single cell. In other words, what would you do if you had a cell and you wanted to record from it? Well, you'd like to understand first and foremost, before you did anything else, you'd like to understand if it's a neuron, what are its firing patterns? How often, what is the structure of its firing? Does it burst? Does it spike? What does it do? Then you'd also like to know its structure. What's the shape of its morphology? Is it branching? Is it tufted, et cetera? And then you'd like to know about its genetics. What's the structure of its genetic program? And it's hard to get all those things at once. Typically, you can get one of those. But there is a technique called patch-seq where it doesn't work in every cell. It has a pretty high failure rate. But if it works, what you do is you essentially record from the cell. Then you take out its RNA out of the nucleus, and then you kind of fill it with this biocytin so that you can later image it. You can imagine all the points of failure in this process. But the thing is, when it works, it's kind of like a Rosetta Stone, because it enables you now to connect data that you may know about morphology with data that you may know about transcriptomics. So we always do a certain amount of this to try to connect data from the different modalities. Oh, the bad thing I was going to tell you was that, the disorganization is that because not one of these archives was capable or willing to host all the modalities, essentially the patch-seq data is spread to the wind. Each of the components of the transcriptomic data goes to one archive, the electrophysiology goes to the other one, and the morphology goes to a third archive. And they are linked in the sense that we know what was what, but still, you can imagine how scary that is. If you want data about a single cell, it's being broadcast to these disparate locations. So that's a bit of a buyer beware, I guess. And we organize data by its structure, right? We have these different levels of data, from raw data to kind of more annotated data to data which is we understand something about it. And so all our data sets, if they're analyzed, are categorized in this way and can be accessed in that way. And we're promoting, fair data sharing principles wherever possible, trying to get standards and resources well identified and things. And so we have Marianne Martone and others who are part of our kind of work are actively involved in the protocols and the distribution of those. We wrote this up sort of motivated by, there was a paper back in 2011 which was written basically about the ENCODE, ENCODE was the profiling of the DNA regulatory elements that came right after the Human Genome Project. And it was, it's a program which is kind of winding down, but it was for many years a really very, very big kind of high throughput kind of thing, which people were collecting all the attack-seek data, different epigenetic data all over the mouse brain more and less in the human. But sort of motivated by this, we wrote a paper to describe the BICCN kind of ecosystem and published it also in PLOS Biology. And you can look at this paper. A lot of what I said and a lot of the infrastructure for what I've said, you can see in that paper. You can read about it right there. So if you are interested in pursuing that, I would say just look that paper up and you can follow up with some of this stuff. Now, and so what we build, we have built applications that I'm pretty sure Rachel will talk about also, something called the Cell Type Knowledge Explorer, where we have this data is kind of in this primary motor cortex was basically organized and cross-referenced and all in kind of one place where users can explore it and learn about the cell types. And I know she'll talk more about that, so I don't have to go into that more. But this is in some sense a kind of a knowledge repository for this data. I mean, ultimately you'd want to have this like in a more expansive knowledge graph environment, something similar to like the eBrains kind of infrastructure, which the European Brain Initiative did a lot of excellent work on the organization of large-scale kind of neuroinformatic knowledge bases, and our infrastructure is not nearly as developed as that, but I think that this is in the spirit of some of that work. And of course, if you're building a knowledge base of this kind of stuff, these are the kind of things you'd want there, right? You want to connect cell types with anatomy, with the genes that are active, and you'd want the annotations and to go back and forth and understand the coordinate systems. And these are all the things that would matter in a knowledge base of cell types of the brain. So one of the things, a little bit of it, one of the things that was really, all these groups were funded in BICCN. And what happens a lot of times is that when these awards are given out, the program, it's probably the same in Canada, similarly to the US or other places that I know in Europe is similar. But these awards are given out with they're given out with the intent and the hope of collaboration and the intent that people will work together in an integrated way. But in practice, it sometimes doesn't work out as perfectly as you would imagine. You would like to kind of do that. But due to, individual organizations, they develop their own kind of vision and agenda, you end up sometimes with multiple data sets that need to be kind of reconciled after the fact. And so in the case of the mouse, this award, we ended up producing, the consortium ended up producing two kind of major atlases of the mouse brain. One is our atlas, this Allen Cell Atlas, and the other came from the Evan McCoskell lab at the Brain Institute, who used the drop-seek technique, a slightly different technique than we used, and then another type of spatial technique for looking at data, and produced another full-scale atlas of the mouse brain. And so, of course, the NIH and people, what we want to do is we want to reconcile these two. We want to understand, which one is correct, or did they reinforce each other, or how do we do that? And so what we're doing now is we're trying to produce a kind of consensus atlas of that. And that would mean, standardized processing, developing a consensus taxonomy, a common annotation, and various atlas tools that support the integration of these data sets. So you got to map, just kind of technical here, I won't go into this too much, but the idea is that mapping, developing techniques for actually, have I counted the same, if something is expressed, if we're saying that a gene is expressed in one, well, what was the process that was used to determine those counts, and why do you say that was expressed? And we need to kind of match that into, if one is done based on 3' counting in the genome, the other is based on 5', we need to understand how they reconcile. And the really way to do that is to reprocess all the data in a common pipeline. We've hosted lots of workshops on this, trying to understand some of these different things. We've probably done 4 or 5 of these workshops over the last few years, in which people came to talk about the process of how about we do that. And it was a very good educational process for everybody. In the end, the goal, of course, is to get some kind of consistent atlas that now we can be consistent across data sets, mapped common across species, enable a kind of organization to understand its anatomy and a spatial distribution of cell types. And we have a mechanism here where now users can map their own data to these atlases and to understand how does the data I have, what cell types exist in the data that I have. I think maybe Rachel will talk about map, we call it Map My Cells, and she'll show you how to use that a little bit later. But it's a very good utility. We weren't the first one to do this, but we've now incorporated several methods into this that you can do this to map your own data. Again, this leads to this nice kind of homology between species across cell types. That a lot of these cell types, they align between, say, marmoset, mouse, human, or macaque. There's a fundamental structure which is kind of preserved across these mammalian brains. You have to get your nomenclature is a big problem, right? Because how are things named? It's just like there's been historically a lot of kind of confusion, or at least complexity, in the naming of organisms and species, right? Early names went before Linnaeus. There was like a pathological naming convention where that thing would be bird with yellow wing that flew by my house yesterday was part of somebody's nomenclature, right? But then Linnaeus kind of established that in something. But this kind of insight is not really fully realized at the level of kind of cell type data just yet, because there's problems of what do they do, what's their function, which can be different in different kind of cases. And we need kind of common annotation to make sense of it. And so we're trying to standardize, we're trying to build software tools that enable the management and the manipulation of these taxonomies so they can communicate. So that if I get a taxonomy of cell types and I send it to someone, another colleague, she can actually, she can ingest it in the same way and begin to work with it. You need proper data containers which do this kind of thing. We have ways of constructing these taxonomies and communicating them. Now, and so a lot of our program consists of sort of outreach. There she is right there. There's Rachel's, her picture. You can see her right there. She's in row 1, 2, 3, 4, 5 over there. And but anyway, she works with one of our groups here that is in terms of in discussing the training and the use and the organization of these taxonomies. And here we built this really kind of cool puzzle that showed the cell types of the primary motor cortex, and it was sort of displayed at a science center downtown in Seattle. And I think that you can learn about it from that. I think we don't have the puzzle with us, I don't think, unfortunately. But yeah, it's too big for the airplane. I mean, you can order it on Amazon, I think, or somewhere. So anyway, now we're moving on. So the next part of this phase of it, remember we talked about these phases of the BRAIN Initiative. This next one is they had to rename it, right? Because we're not going to give the money to the same people. We're going to change the people maybe, and so we got to change the name. And so this was called BRAIN Initiative Cell Atlas Network. It's really the same thing. It's just that now we're going to be doing it more in human. The mouse is, at some point, even though we have this encyclopedia of this data and more or less a description of cell types across the whole mouse brain and a good part of the marmoset brain. and we've prototyped it in the human. But at some point, as people do, you sort of declare victory. Now that we don't know what these cell types do, it's still going to be perhaps decades of work to understand what they mean or how they interact, but you have to declare it complete. And so that is complete, that is said to be done, and now we're moving on to the human, and that's really what the BICAN is. It's now trying to extend this work into human. And this is a whole, there's the arrow again, here we are right here, okay? And this is to go on to about 2026, 2027, and at this point, we'll have to declare victory, because I guess there'll be other activities that will require interest and funding. So this network is a pretty complicated network of this BICAN. This is just showing you who are the kind of players and what does this look like, and how did they set this up? Well, what they did is that on top up there, these things is called UMs. And a UM is a structure, so the convention, you've probably heard of RO1s, right? RO1s are those were the kind of, or R something or other. Those are the awards in the NIH that sort of, they're individual investigator awards that say, you go ahead and you do this work you propose, we think it's good, and get to work, But once they do that and they give you that, they can't really bother you. You have to, in fact, you can now tell them what you think of them until the next time you have to ask for more money. No one can bother you, right? Yeah. Well, so there's a new mechanism called the UMs, or the UOs too, and this is different, right? This mechanism here, and it's not, so the NIH also has these things they call contracts. And a contract differs from an RO1. A contract is that if you say you're going to do something, you must do it, and you must do the things you're saying. And if you don't do them, they're not going to give you the money. Well, a UM is like somewhere between that. It's you are making the proposal, but it enables them to kind of get in there and mess around with your space a little bit more. They can keep asking you where it is, and they're more involved, right? And so these big things, these are big programs, which I'm going to tell you about, are these major profiling for the human brain from Joe Ecker, Steve McCarroll, Ed Lean, Arnold Kriegstein, and these are different kind of major profiling things of the human brain. I'll tell you what their projects are. And then there's these kind of informatics, these sort of U24, and these are our different groups here which are involved in some aspect of the informatics processing, and I'll tell you what those do. Then on the other hand, you have all the R's over here. These are the people that they're just proposing individual kind of things, and they're not looked after quite as closely as the UMs, right? They're left to do their own thing, but they're supposed to be integrating into our world, and similar other things there. So we use a lot of the same infrastructure that we did for the BICCN. It's just gotten more complicated, and I want to tell you about what, and you can imagine doing human why it's complicated, but I'm going to try to explain like some more of that. This ecosystem now for human becomes a lot more complicated because now you have issues of tissue, right? Whereas before you simply had the mouse, you can produce these mouse brains. The mice are sacrificed. You can use them. There's as many as you need, etc. In human, you have issues of tissue availability, issues of privacy, issues of identifiability, issues of size and scale, issues of tissue access, all this in an immense amount of complexities. And all this has to be accounted for. People have to, and when you, it also, it's much less clear when you select data from a given region, are you really getting that region, right? Because it's not, these are not like inbred mice. Primary motor cortex may have a dramatically different extent in the human than it did, between human than it did in the mouse. And so we have a much larger kind of ecosystem with these different kind of components, and a place where investigators now they go in and they actually can sort of visually see an atlas and see tissue mapped to that and sort of try to pick an area from where they want to kind of work and profile, and then the tissue is kind of selected and brought to them. And then there's a lot of work in terms of trying to standardize metadata, different kind of organization toolkits for handling that. We have common processing. In the BICCN, people could generate their own data, and they could run it in their own lab, or they could send it to their own kind of sequencing center they want to. But now, in this new world, the NIH wants everyone to use a set of sequencing centers. So all data goes to these sequencing centers, they produce it, and then it comes back to the archives, and they bring it back. And so this is like kind of the informatics infrastructure of all this here. There's the UMs generating the data up there. There's the data coming from neurobiobanks and specimen cores. It goes to sequencing. It goes to data archives. We have all this informatic structure regarding selection of the data, organizing, sequencing, and so it's kind of big. So this is kind of an outline here of kind of the major projects in BICAN that are going on. Now, don't worry about this slide. It's not. You couldn't process this slide really, just because it would be hard to see what the differences are. But I'm just using this as a kind of a placeholder when I tell you about the individual ones. There's these more or less these 6 different projects that kind of profile different aspects of either development or population or other things in the brain. One of them is, so this is one of the proposals is from this Joe Ecker lab called so these are the big UMs. These are the major data producing things of BICAN and how they're going in. And you know, this is the data that, for example, some of this we may be able to identify, you know, and to understand how this might relate to BigBrain and how we might be able to, you know, identify synergies here, which would be interesting and valuable. Here they're doing an epigenetic profiling of up to like 200 different regions of the human brain, according to an ontology which is common between us, our work at the Allen and work that they're doing there. They're doing attack-seq and methylation studies across the brain, generating those sequence data for that. Arnold Kriegstein from San Francisco UCSF is making a human developmental atlas over, they're actually doing 3 species and 4 different development stages and kind of, you know, using different kind of techniques to kind of put together all this to produce a kind of developmental series. Steve McCarroll and Evan McCosco are doing a population study. Here they're looking at 50 different brain areas in up to 200 kind of adults using so-called multi-ohm technique. And multi-ohm is a technique where now you can actually, instead of just doing the single cell sequencing in a cell, you can actually run the attack-seq or the epigenetic thing in the same cell. So you get actually both the regulatory information and the gene expression in the same cell. With all these things, you want to always understand with all these that every time you add one of these modalities, there's a loss of resolution. You don't get, it's not like you're getting everything you got from both into one. You get a reduced resolution, more failure rate, but you get a thing. And also in single cell technologies, people have opted, you know, they've opted for, of course, cheaper methods that can do higher throughput compared to, say, some of the original things that in earlier things that were used a little bit less, such as the SMART-seq, and those were extremely robust, and they were produced really, really accurate numbers of counts of expression in the brain. With things like 10x genomics and more recent methods, they're much cheaper, they're much higher throughput, but there's a loss of resolution. They're quite good if your goal is to characterize what's different between cells or characterize a cell type. Where they're not so good is that I'm interested in Parkinson's disease, and I need to know what's really happening in this cell. And there, you know, then you have a loss of resolution, which would really be, you know, important to the cell dynamics, and you need to investigate much more closely. Paula Orlota is doing a very extensive developmental atlas of the mouse brain in this thing with about 15 or so different, or to 20 different regions. We've just actually published some of this data with her, not published, it's in review now. But this is a very fine, detailed atlas of the mouse brain. Another atlas from Changhuang Liu at San Diego, connecting in development, connecting methylation and transcriptomics. And finally, one I'll spend just a few more minutes on here is from our group with Ed Lean, is a kind of functionally guide, and this is one that may see some of the most synergy with some of our work here today, is that this thing that what we call the human and mammalian brain atlas. And this is the idea that not all these pieces are in the works just yet, but the goal would be to try to understand, is there a connection between, functional identification of brain regions has come, say, from fMRI, either, default network or task related, and actual cell types of the brain. Can we identify in a correspondence or understanding of cell types active in different functional brain regions? And so this front end of it is not operational just yet. We don't have the in vivo kind of scanning part that is connected just yet. That's a part of the pipeline that's still under development. But we have now done we are, as a group, doing a very deep dive into the basal ganglia across human, macaque, and marmoset to understand what is kind of the cell type structure of those. And the goal is to kind of build a similar atlas for the, this is our so-called, just like we called the mini atlas of the BICCN, the mini atlas here is this basal ganglia atlas for the human. And this is basically it. It's uses kind of similar techniques to the kind of things that were developed in kind of using some of the technologies of the big brain and other kind of atlasing things. And the goal is to produce this kind of data product, which enables connecting anatomy with cellular structure and understanding of the function of cell types. Here, like I said, we're doing this deep dive into the basal ganglia across these 3 species. This is a really good area as a prototype region because it connects several kind of different, functional system areas of the brain, including, the globus pallidus, the caudiputamen, et cetera, substantia nigra. And we've actually done really pretty well with this. We really have a really kind of a lot of data collected with this. We've mapped all this data together. We understand kind of the taxonomic structure, and we've characterized a lot of the cell types in these different things, and they've been mapped with good, strong spatial data. And so this is producing like a first level system that kind of is really a kind of our first mini atlas, if you will, as part of the human studies. And again, the goal, we get this kind of aligned taxonomy. Here's just a taxonomy showing major cell types in the basal ganglia, and how the clusters map between, let's say, literature identified or those cell types that have been identified in the literature versus those that we found in the data in macaque, those that we find in human. We link this with kind of cellular ontology and different kind of informatics tools that are known in the field. We have working with people like David Somi-Sutherland from the Sanger, who's very involved in cell ontology, to try to come up with good nomenclature for this. And it produces a nice kind of taxonomy of cell types where you can actually map and visualize and identify common clusters between the species and what they do and what they mean. We find kind of conservation of these cell types, right, between macaque and human pretty well, but different groups, these medium spiny neurons and granular cells, you can see that there's a good kind of, the numbers or the percentage of these existing in the dorsal and ventral striatum are pretty similar. And the different subtypes are really kind of identifiable in both data sets and kind of corroborated when you use the spatial data to the MERFISH data. The data reflects the right connectivity between these different regions of kind of the brain, from the sensory motor areas and the limbic areas. Where the cell types are active, we know are corresponding between the right kind of areas of the brain. So it does kind of make sense as a description of basal ganglia. And we get this nice kind of spatial distribution with all the different kind of gradients in the kind of the striatum, with all these different areas of the core, the matrix area, and all the different, the shell, and all these different important areas of the basal ganglia. So this is kind of becoming like it's a proto atlas of the first kind of version of this. Now, an atlas as part of the BICCN was done. A first kind of human thing was done by Sten Linnerson and Kim Stiletti, and they did a kind of a coarse atlas of profiling different regions of the human brain. And so we use this. We've kind of got this data in our Allen atlases, and we can map to it and kind of keep things kind of organized in that way. Finally, just very briefly, I'll just mention another thing on a disease focus that we're doing is we have an effort which called the Seattle Alzheimer's Disease Brain Cell Atlas. And I have just a couple more slides and I'll be done. But this is kind of it's an effort to use a lot of these single cell kind of techniques here to investigate neuropathology. We have some working with Dirk Teen from the University of Washington and an ADRC kind of Alzheimer's disease research kind of center there to actually get kind of tissues. And what's really neat about that is that the data comes from the Kaiser Permanente kind of study that they had something they called the ACT, or the Adult Changes in Thought. And basically, if you're enrolled in the Kaiser health system, you have the option from age 50 to join into this kind of periodic thing. It's a service provided where they basically just keep track of you with blood work. Every 5 years, you get a brain scan, you get different things, and they track people to see. It's a data collection things, and some will develop neuropathologies and dementia, and those we have all the metadata now associated with that. So all that metadata is what has gone into this study. And then we can do the molecular analysis and the spatial analysis and try to pull it all together. And then we have a really strong neuropathology core involved in like quantifying what different kind of structure plaques we see in the data. And there's a viewer built for it. I know I'm running out of time, but so where this connects back with the other work here, so I want to identify is that now, we had the taxonomic structure of cell types in where this data came from. In this case, all this data came from the middle temporal gyrus in the human. And that was the first area that we looked at. And we're looking at some other areas, but this was the first area we looked at. And we basically started with that taxonomy that we had already developed in the human from middle temporal gyrus. And now using all these, the Alzheimer's donors, we can investigate the cell types there and extend this taxonomy to understand, are there any modified cell types? We use the mapping tools to see, has anything changed? And is this kind of, and how does it look? And so, using this way, we get kind of a map of how cell types are changed, what are vulnerable cell types, and how are they changed over the progress, the pseudo-progression of disease in individuals. And this is all available through an open kind of resource. At the CAD, if you go to look it up, cad.org, you can see all these different tools and all these different kind of things, and they're all integrated with some of the other resources that I said. So I think is that it. I think I'm more or less on time. But it's just a summary, a few points here. Molecular profiling of cell types really enables the development of these comprehensive atlases of the mammalian brain in mouse and macaque, now in human. This BICC and BICAN was a large collaborative effort in brain atlasing to try to profile these different kind of cell types, to understand what are the cell types and their maps and their connections. And a substantial infrastructure has been developed, sort of parallel to work that's gone on in Canada, as well as in eBrains in Europe. And we should look to understand over the next few days, how does this work synergize with BigBrain, and how can we bring some of these opportunities to closer to where we all want to go. So thank you very much.